Hello and welcome to the big old Chapter 8 lecture, Energy in the Cell and Enzymes. There's going to be a lot of abbreviations in this one, just to keep the size of the sentences down, so hopefully you're taking good notes from the sound of my mellifluous, melodious voice. So a quick slide of abbreviations. If I have a down arrow, that's a downhill. Up arrow, uphill, not rocket surgery. Delta G is a term for what's called free energy. Free energy is energy that does work, or in other words, makes physical changes. E, you'll see all over the place, that's just energy. PE is potential energy. RXN is reaction. Endergonic, which is a reaction that requires work, is Ender-G. Exergonic, which is a reaction that produces work, is Exer-G. So keep up, basically just a way for you guys to know what I'm talking about in all the shorthand. First off, let's talk about how metabolic pathways are all about managing energy in the cells, right? So what you have are a number of chemical reactions. A metabolic pathway isn't just a single chemical reaction. C6H12O6 plus 6O2 yields 6CO2 plus 6H2O and ATP production. It's written as one reaction, but it's actually a large series of steps. And the idea is that every single reaction releases energy, and you lose some of that energy in various forms. Light, heat, sound, depending on what kind of reaction it is. And so to minimize energy loss, you rebind up the products. So instead of having C6H12O6 plus 6O2 yields 6CO2 plus 6H2O, instead of just one big step, you go C6H12O6 broken down into 2C3H3O3. And that way you lose some energy in that chemical reaction, but before you lose everything, you bind it back up into the chemical C3H3O3 because those bonds store energy. So all those steps help us from losing energy. And then to minimize the amount of energy that a metabolic pathway costs, we have enzymes. Every single chemical reaction in the metabolic pathway is catalyzed by an enzyme. Catabolic pathways are pathways that usually break molecules. They break bonds. And whenever you break bonds, you release the energy those bonds stored. So you get a net energy production from catabolic pathways. Cellular respiration is one large, complicated catabolic pathway that ends in binding up the energy produced into ATP. An anabolic pathway is building stuff. So these are chemical reactions that require energy. We often call this the biosynthetic reactions. In other words, we're building stuff. So for instance, macromolecules, building any macromolecule is definitely biosynthetic, definitely anabolic. Proteins, for example, are a complicated polymer, right? First off, you have a bunch of amino acids. And you need to link those amino acids together in single covalent bonds to make the polypeptide. And that's primary structure. Secondary structure, some hydrogen bonds forming. Tertiary structure is, again, covalent linkages forming between different parts of the polypeptide to bind it closer together. And then that's tertiary structure. And then in quaternary, you end up binding multiple tertiary polypeptides together. So this is costly. It takes energy to form all these bonds. And that energy, it comes from the catabolic pathways. So generally, building anything from monomers into polymers is biosynthetic. Uphill and downhill are common terms in this chapter. Downhill reactions simply release energy. If you're riding a bike and going downhill, you're generating velocity. You're not having to put your own energy into it. You're gaining energy by going downhill. It's a terrible metaphor because there's gravity, energy, and all that stuff in there. But downhill is a downhill reaction releases energy. Consequently, an uphill reaction requires energy. So you're riding your bike uphill, you have to pedal. You're putting energy into it. Uphill reactions require energy. Downhill reactions release energy. And this gets all into bioenergetics, which is the study of energy flow in biological systems. And what we're primarily going to be looking at is how energy coupling works in biological systems. Basically where we run a catabolic pathway in order to have energy to run an anabolic pathway. We'll talk a little bit more about that a little bit later. But first, let's define the different forms of energy. Yeah, we're going basic here. So, kinetic energy. You'll see I had a little bit of fun with my animations. Kinetic energy is sliding in. Kinetic energy is energy produced in movement. So, movement is kinetic energy. Thermal energy is energy in the form of heat. Kinetic energy is energy in the form of movement. Thermal energy is energy in the form of heat. Potential energy. This is an interesting concept. So potential energy is basically the energy that is potentially available in a system due to its physical structure or position. Right? So if I'm holding a marker here, I have a good grip on it. It's pretty stable. It's got low potential energy in this system. If I hold it here, I've got a poorer grip. There's higher potential energy. And then if I try and balance it on my thumb, that didn't work at all. There was very high potential energy. So position can produce potential energy, and at the same time, structure can also produce potential energy. An unstable structure has high potential energy. Nitroglycerin, kinetic energy can cause an explosion because nitroglycerin is so unstable. So that obviously holds a ton of potential energy. So energy possessed by matter due to location or structure. And when we talk about chemical or potential energy, we're going to be talking about chemical energy, which is potential energy available for reactions from the chemical bonds. So the way the bonds are in the molecule determines the amount of potential energy available for chemical reactions. Like we talked about with ATP, because those phosphates are negatively charged and trying to repel, and because the bonds between the phosphates are those extremely weak phosphate bonds, you've got a structure that's trying to tear itself apart at the seams and is highly unstable, so it has high potential energy, it has high chemical energy. So there you go. Whenever we talk about energy, we want to keep in mind the good old laws of thermodynamics. The two basic laws of thermodynamics. 1, the first law governs energy transformations. Basically, we say that energy can be transformed or transferred, but it can never be created or destroyed. This is the conservation of energy. Energy can be transferred, right? So here's kinetic energy in my hand. And here's my marker. I'm transferring kinetic energy from my hand to the marker, and the marker's moving in response. And at the same time, I'm transforming some of that kinetic energy into sound energy. So we have kinetic energy, chemical energy, sound energy, gravity. There's lots of different forms, gravitational potential energy, lots of different types of energy out there. But energy is neither created nor destroyed. The energy in the system, in the universe, is all the energy that will be in the universe and all the energy that will ever be in the universe. So we're not creating new energy and we're not destroying energy. So when you have a chemical reaction that produces a lot of energy, it's not creating it out of thin air. We're converting potential energy into light, heat, sound, various types of energy. So transferring, transforming. So the same energy that was in the dinosaurs exists in the universe now. The same energy that is in any living thing, right? It will never cease. It will never go away. Energy is never created and never destroyed. That's about as close to a religion as I have right there. So, the second law of thermodynamics deals with entropy. So let's describe entropy. Entropy is the tendency of matter to move from states of order to a state of disorder. What this means is that any organized, ordered system over time will lose its order. Things break down, right? Metal rusts and breaks down. Mountains grind to dust. Everything breaks down. Entropy is unavoidable. Matter moves from a state of order to disorder. The second law of thermodynamics simply states that entropy is the result of all energy transformation and transferal. In other words, we do not have infinite recycling of energy. So, say I start a marble rolling. On a flat surface, the marble will slowly come to a stop. This is because the marble rolling is an organized, ordered system, and entropy comes into play as it loses velocity, and then it comes to a stop. The energy that was propelling it kinetically is lost. Entropy. If we could recycle that energy infinitely, I'd just need to push on the marble, and it would continue rolling no matter what. So, a lot of people out there in the past have attempted to violate the second law of thermodynamics by getting a system that produces more energy than it starts with. So, you put, if it's a rolling machine, you roll it, and the initial energy you put into it is not lost, and it just serves to continually accelerate the object, right? We know that's not real, but a lot of people have tried to design something like that. It's a perpetual motion machine, which leads to one of my favorite physics jokes.
I know. And this perpetual motion machine she made today is a joke. It just keeps going faster and faster. Lisa, get in here. In this house, we obey the laws of thermodynamics.
Anyway, just because Simpsons are great, or at least old Simpsons are great. New Simpsons is dead to me. All right, let's talk about biology and thermodynamics. Cells are engines of creation. They synthesize phospholipids, they synthesize macromolecules, they synthesize DNA, they synthesize RNA. They're constantly producing, constantly creating order. So why don't cells violate the second law of thermodynamics? Well, cells contribute to entropy because they must break down the nutrients they bring in. And then they must break them down in catabolic pathways. So, for every bit of energy devoted to increasing order, cells must first destroy order in order to harvest energy. So cells are harnessing entropy in order to create order. And we know cells don't have an infinite lifespan. So eventually, even the cell succumbs to entropy in total when it dies and breaks down. So, cells don't violate the second law simply because of all the destructive chemical reactions occurring with the cell and the fact that eventually they die. All right, as we're getting into biology stuff, let's talk about reactions. Spontaneous reactions. This is a term for a reaction that seems to occur on its own without any energy. So a reaction that doesn't seem to require any energy to occur. Now, the thing about this is that all reactions require energy to occur. Every single reaction requires energy. So a spontaneous reaction may look like it doesn't need any energy. For instance, if you combine baking soda and vinegar, you'll get bubble volcano, and that seems spontaneous. You certainly didn't add any additional energy into the system. The truth is that it still used energy to start the reactions. It's just that a spontaneous reaction occurs with simply ambient energy. The energy that's just immediately available in the environment is enough to trigger the reaction. So it seems like it doesn't need energy, or at least it doesn't need additional energy to occur. So that's a spontaneous reaction. I used to just tell people it doesn't require energy, but that's not technically correct. And I want to be as pedantic as possible because this is science, and my identity is being a smart person, and so I have to be ultra completely smart and correct. And now I can have people correct me on every little thing I ever get wrong. So that was a mistake on my part. As I said, all reactions require energy. A spontaneous reaction just needs the ambient energy in the system, so it doesn't appear to require any, or it doesn't require any additional energy. So cells, in their biosynthetic pathways, require work energy to build stuff. Work is a kind of energy that basically describes the energy used to make physical changes. If you are hanging out with your friend going up and down on the seesaw is using work energy to change its position. It's physically changing its position in space, and that requires work energy. Moving your muscles and your bones, that's work energy. So the energy available for work is free energy. Free energy is abbreviated with the good old delta G. So all work energy goes into reactions that make physical changes, and those reactions move towards stability, a stable system where no more energy is being used. So an unstable system has a higher potential energy, and that higher potential energy means an unstable system will produce more work energy. As that energy is produced, we have a reaction. That unstable system cranks out a bunch of energy, and the reaction moves towards stability. For instance, if you take hydrogen, 2 hydrogen, and combust it with 1/2 oxygen, 1/2 O2, because oxygen never exists as single oxygen atoms. It's always O2. So 2 hydrogen plus 1/2 O2 yields H2O, right? H2O is a nice, stable, polar covalent molecule. And the reaction is explosive. It's combustion. It produces light, heat, sound. So it's this hydrogen and oxygen is a very unstable mix, and it creates a very violent reaction that releases a lot of energy, but the ultimate product of that is something that's very stable, water. So a stable system has the lowest free energy that you can get from breaking it down. A stable system has a very low potential energy, and it costs more energy than it's worth to break it down because you're going to get very little, if any, free energy. If you try and break down a stable system to try and get energy out of it, you're going to be out of luck, really. Anyway, so spontaneous reactions generally involve unstable reactants. So these are reactants that are prone to changes through chemical contact, right? For instance, acetic acid, vinegar, kicks off protons. And baking soda can end up releasing OH that binds to those protons, and you get water, and then you also get a carbon dioxide gas. So just by virtue of producing, because acetic acid is in water, it's kicking off protons, and when you put it in with that baking soda, the OHs are kicked off, and you get OH reacting with H for the protons, and you get water, a nice stable molecule, and as a byproduct of that unstable system, you also get carbon dioxide in a really pretty chemical reaction, especially if you add some orange food coloring to it and a paper mache volcano. So spontaneous reactions are what you want, or at least something close to spontaneous, when you want to generate free energy. So equilibrium is the result of a reaction that produces free energy, and we move to a stable system. The system produced through that reaction that has maximum stability. This is not great for metabolism, right? If we want to harvest as much energy as possible from glucose, we don't want just one big chemical reaction. We're going to have a bunch of enzymes around that are going to reduce the energy required to start off our metabolism reactions, to start digesting glucose. We're going to kick that energy low enough that we can set off the reaction. And it's exergonic, exo, outside, gonic work. We're producing work, releasing work energy, increasing delta G. An exergonic reaction occurs when you have reactants that have high potential energy. They tend to have a spontaneous reaction, or if you're lucky and you have a really good enzyme, the enzyme essentially makes it spontaneous, and you get positive free energy. You increase the amount of free energy in the system. This is, of course, a downhill reaction. The problem being that if you just do this all at once, you're going to lose a lot of your energy to transformation. So instead, we have a number of spontaneous reactions, spontaneous due specifically to the enzymes involved, right? There's actually a series of steps. It's all kicked off with a little bit of energy at the start. The very first reaction, where we take that C6H12O6 and crank out 2 of those C3H3O3s, that actually requires 2 ATP to get started. So it needs energy to start. However, it's can be considered spontaneous to a degree because we actually get 4 ATP out of it. So we get more energy out of it than we had to put into it thanks to our pretty happy enzymes. So glucose is our initial reactant, and we run glucose through a large series of steps to minimize the amount of energy transferal to transformations to other waste energy, temperature, light, heat, heat, light, sound, that sort of thing. We want to minimize that. So whenever we have that reaction go off, we quickly bind it up into something. And then that keeps us from losing all the energy, binds up all that energy that was going to be lost into new bonds, chemical energy, and then we can take that product into a reaction, and boom, and then bind it up and react and bind it up and react and bind it up. And that way we can produce what we need out of these chemical reactions without losing everything and being incredibly wasteful. So each one of these steps is exergonic. Each one of these steps is increasing free energy in the system. The reaction product in every single step is the reactant of the next step, right? So C6H12O6 reacts, and we break it down into 2C3H3O3. Well, 2C3H3O3 are broken down into C2H3O? Yeah, I think that's what it is. I'll know for certain in the cellular respiration lecture. Anyway, trying to, off the top of my head, probably wrong. Who knows? Anyway, C3H3O3 was the product of the initial glucose degradation reaction. And then it became the reactant of the next reaction. So the reaction product is the reactant in the next step, and this also does us the solid of never reaching equilibrium. So we don't lose all the energy as we hit equilibrium. Instead, we save some energy into the C3H3O3, the pyruvate, and then when we break down the pyruvate and release energy, we quickly bind it back up and save some of that energy into the acetyl. And then the acetyl goes into a reaction in which it produces oxaloacetate or something like that. Anyway, getting ahead of myself. These steps prevent immediate reaching of equilibrium and loss of all our energy. So, enzymes catalyzing every single one of our respiration reactions means we have a high potential energy system in metabolism. Because we have a high potential energy, so the enzymes reduce the amount of energy needed for these reactions to fire off, right? So by reducing energy needed, we basically increase potential energy available when the reaction occurs. So enzymes catalyzing every single one of the reactions in respiration, cellular metabolism there, means high potential energy in the system, so that when the reactions occur, we get a large increase, a positive increase in delta G, free energy. Glucose is made of covalent bonds. Energy is required. So the term here I want to put in is that we get net free energy out of the metabolic pathways. It's always going to need energy to start any of those reactions. We always have to put energy into the reactions, no matter what reaction it is. It needs energy. So when it's exergonic, that means you're getting a net increase in free energy. Bonus to free energy, even after you spend energy to set off the reaction. And as I said so many times now, the stepwise reactions are there to conserve potential energy, so we don't immediately convert all our potential energy to the waste energy and crap out into some stable system before we can get as much energy as necessary for making our ATPs. So the chemical bonds reduce energy loss to waste, to transformations into light, heat, sound, etc. So, yeah, said that like 8 times now, but I guess maybe it's just important to know. So, energy released as free energy can be harvested for work. That work is generally making the ATPs or powering the next chemical reaction. Ultimately, we use the free energy from our exergonic reactions for our biosynthesis pathways, our anabolic systems that are endergonic. ATP is the ultimate storage of potential energy because the molecule itself is just so damn unstable. So, ultimate result is that glucose hydrolysis, starting off all the steps of cellular respiration, gets us to ATP. Endergonic reactions are obviously not spontaneous because they require work energy. So the energy required to start the reaction is much greater than the energy that might be produced in the reaction, if any is produced at all. So, in other words, you got to spend energy for an endergonic reaction. And that energy is in the form of work energy, delta G. These are obviously uphill reactions. So, you have the exergonic reactions in cellular respiration that give us free energy. And we take that free energy, bind it up into ATPs, and then send those ATPs to help us perform those endergonic reactions. And coupling an increase in free energy to using free energy is called energy coupling. Yeah. So, energy coupling is all about taking the energy produced through metabolism and spending it to make stuff, biosynthesis. Good view of this energy coupling is in good old-fashioned plants. So what process makes food for the plants? It's endergonic. It starts with a P and ends with photosynthesis. Yeah, that's right, photosynthesis. 6CO2 plus 6H2O yields C6H12O6 plus 6O2. This is the chemical formula for photosynthesis. You'll note it is actually the reverse of the chemical formula for cellular respiration, which is just pretty cool on its own. Anyway, what's the energy source for photosynthesis? Yes, kitty. What's the energy source for photosynthesis? Yeah, the sun. Yeah, you should have known that. If you didn't know that, then I weep for your science education. It's the sun, people. Photosynthesis is sunlight. You terrible idiot straw man student I just created. So why don't cells reach equilibrium? Well, a cell at equilibrium, where we have stable low potential energy, is a dead cell. That cell done died. That is why it is not at equilibrium. So cells are constantly bringing in new molecules through endocytosis, nutrients that can be broken down for more reactants, and we're constantly getting rid of stable wastes. So to prevent a buildup of those stable non-reactive wastes, we exocytose them out of the cell. Get rid of the wastes that are stable, bring in new unstable shit. And this prevents us from ever reaching equilibrium until your cell sort of hits its lifespan's end and dies anyway. Yeah. So, endocytosis brings in new reactants, that allow us to do downhill reactions that give us free energy, and each product is thrown into the next step to become the reactant, and thus we... Okay, cat, come on, I have to swaddle you. Yes, 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 I know. But if you freak out, you'll get another seizure. The products of the reactants of the reaction become the reactants in the next step. We hit that already, and we just continually prevent total energy loss, because when we lose all that energy, we hit our stable state. So in preventing the accumulation of our end state product that's stable, we prevent equilibrium. So, yeah, even though C3H3O3 is a nice, it would be stable, you then have an enzyme to crank down the energy we need to break that down, and we break it down into our C2H3O or C2H2O. Maybe it's C2H2O. Anyway, whatever, acetyl. So, we prevent equilibrium. Exocytosis is the other way. By getting rid of our stable waste products, we prevent equilibrium. Cells try not to die. That is your take-home message right there. Okay, so now that we have all this free energy available from our ATPs that we've painstakingly made, how can a cell put work energy to use? Chemical work is one way. So we've already painstakingly said this: biosynthesis pathways, making new chemicals through endergonic reactions. Transport work is another way. The Chapter 7 lecture had us talk in length about active transport proteins and how they need the ATPs because they violate the natural laws of diffusion. And so we can just pump energy into it in order to have that happen. So transport work is cellular work. It's work energy. So active transport uses work energy. Mechanical work, such as the elements of the cytoskeleton that move around. So just, breaking down and rebuilding cytoskeleton in new places, that's mechanical work. Your cilia and flagella are mechanical work. Even your big, large muscles are mechanical work. So basically, almost everything a cell does everything a cell does to function and be alive takes work energy. And it's ATP that is nice and unstable, having that high potential energy so that it can release a large amount of work energy that is going to be used to power all that work. And I literally think I just said this slide. Energy coupling. Wow, I did that again. Exergonic coupled to endergonic. In cells, it's ATP. Break down to make free energy. Oh, God, this is where I talk about ATP again. The repetition's important. I'm just tired. So what's the nitrogenous base in ATP? Huh? What's that? What's that? A. Starts with A. Ends with C. Sine. A blank sine. Middle we have dino. Adenosine. I hope you got there before I did. The sugar is good old ribose. Oh, well, adenine is the nitrogenous base. Bound to a ribose makes it adenosine. And then we have 3 phosphate groups. And those are held together by those extremely weak phosphate bonds. Those phosphate groups carry a negative charge, which means each of those three groups are trying to repel each other, and they are being held together by an extremely weak bond, meaning ATP is highly unstable. So it has a high amount of what that's locked up in those chemical bonds? Starts with P. Ends with potential energy? That's right, potential energy. And when we break ATP down, it yields a high amount of, or an increase in, work energy. Yeah, you got there. You got there. Okay, so we break ATP through hydrolysis, and we cleave off one phosphate group. When we break ATP to make use of that unstable molecule's high potential energy, we bust off one phosphate. So ATP is hydrolyzed. I really shouldn't put plus H2O there. There's no good way to abbreviate H2O there, because when it's plus H2O, that says that they're reacting together. Where in reality, the H2O, the hydrogens and the oxygens are not incorporated into the products. Because when you have two reactants, you're going to have, all the ingredients on the left side on the right side. H2O does not get incorporated on the right side, so that plus is crap, but there's nothing good in PowerPoint to denote when you add water to break bonds. But that's what we're doing. We're adding water to break the bonds between two of the phosphates. So we bust off a phosphate, and that's where that plus P is. And what we're left behind is adenosine and two phosphates. I wonder what that D stands for. Di. Adenosine diphosphate. So when we hydrolyze ATP, we knock off a phosphate, we get adenosine diphosphate. Obviously, this is exergonic, meaning it releases what now? Yes, work energy. Good. Need some water. It takes a surprisingly large amount of ATPs to power a cell. A cell has vast needs of ATP. So a cell does not exocytose adenosine diphosphate and that phosphate we cleaved off. It doesn't go, done, kick them out. Instead, it uses one of two basic processes to react them together and turn it back into ATP. So we have substrate level phosphorylation, in which you use a little bit of energy and directly bind the phosphate to the ADP to create ATP. And you have oxidative phosphorylation, which involves a fancy enzyme called ATP synthase. Either way, the whole idea is to recycle the ADP plus P into ATP. So before recycling, that phosphate group binds to whatever does the work. So I said that in the Chapter 7 Lecture 2, but I can't say it enough. When we cleave off the phosphate and release work energy, the phosphate binds to whatever's doing the work. And the work is done, and then the phosphate is released. So that thing doing the work that gets the phosphate between the ATP into ADP plus P and the ADP plus P into ATP is called the phosphorylated intermediate. Phosphorylated intermediate is what's doing the work, and it's obviously getting the phosphate. So the phosphorylated intermediate needs free energy to do its work. So when we hydrolyze ATP, that loose phosphate that we busted off binds the phosphorylated intermediate and brings with it the energy necessary to do that work. And when the work is finished, the phosphate is released, and then that's when we bind it back up with ADP and get our ATP again. So we recycle. Without recycling, well, bad shit happens. The human muscle cell uses all of its ATP in less than 60 seconds. So as soon as you start contracting, in less than a minute, every single ATP molecule in that cell is used up. And if you don't have additional ATP, then you're done contracting. So without recycling, we would have to produce our entire human body weight in new ATP molecules every single day just to power the energy requirements of our body. That is impossible. We would die while eating trying to get enough nutrients, but the very energy required to break down the nutrients to make ATP would take more energy than we have available, and we'd all die with cake in our mouths, probably naked on the floor weeping. Yeah, that was disturbing imagery. I would produce 1.08 times 10 to the 26 molecules every day, 108 trillion. That's not exactly a number you can just internalize. You go, oh, okay, sure. Means nothing to you. But anyway, we can recycle 10 million molecules of ATP per second per cell. So in 1 second, a single cell will use 10 million molecules of ATP and then regenerate those 10 million molecules. We're not violating the first law or the second law of thermodynamics. We're not perpetual motioning here because this recycling requires energy. And so that's the whole point of metabolism. The cellular respiration where we break down glucose to get the energy to make ATP, right? If we don't have glucose coming into the system so we can break it down to get energy to recycle our ATP, then you spend your ATP and you done died. Oh, it's bright. It's so bright. Okay, my eyes have adjusted. Part 2 is where we talk about all those beautiful enzymes I've been mentioning. So an abbreviation I'll use a little bit around here is activation energy, E sub A. This is literally the energy required to start or activate a chemical reaction. The energy required to activate or start a chemical reaction. Energy required to make or break a chemical bond. So, activation energy is present whether it's an endergonic reaction or an exergonic reaction. They all have an activation energy, right? And the activation energy controls how often these reactions are going to occur. So, say we have our graph here, right? And this is our energy, kilocalories or joules or some shit. We'll just be like super vague and call it units. Oh, I love writing with the mouse. So, there's our standard energy units. So this is the energy. I lost my thing. This is our energy needed to start. Our cell can only devote so much energy. So say our cell has 10 energy per second to devote to this particular chemical reaction. Maybe it's the chemical reaction that makes you think happy thoughts, right? So maybe perhaps on its own, this chemical reaction requires 5 energies to start, right? So you need 5 energies in order to fire off that chemical reaction. So if you get 10 energy per second, how many times can you do it? Oh, you can do this reaction twice per second. Oh, not very often. You're not thinking very good, happy thoughts. So then you have an enzyme that comes along and it says, hey, buddy, don't mind if I help out. And the enzyme reduces activation energy. Now I can fire that off with just 2 energy. And suddenly we're up to, where's my thing? Come back here, thing. So bright. Suddenly we're up to 5 reactions per second. So, yeah. But, well, wrong button. Come back. This button. That button. Haha. And then this button. I can click again. So an enzyme is what's called a catalyst, specifically a biological catalyst. So an enzyme falls under the heading of catalysts. Any catalyst reduces the activation energy of a chemical reaction. It makes the energy required to start it smaller so that it can go off more easily or more often. An enzyme is a biological catalyst. So it increases reaction rates, but when we say that, we want to be nice and specific and pedantic. So in this case, so some people, when they see reaction rate, might think of velocity. They make the reaction occur faster or that it takes less time. Like say it took 2 seconds to break glucose into pyruvate. And they're like, oh, it increases reaction rate. Now it only takes 1 second to break glucose into pyruvate. It's more like, no. It's more like you can break glucose into pyruvate more often. So it still takes 2 seconds in this metaphor, but you can do a lot more glucose into pyruvate breakdowns when that catalyst is around than when it's not. Like if our graph we just did was for that, you can do 2 glucose per second broken down, or in the presence of the enzyme, 5 glucose per second. So, increases the reaction rate, specifically how often the reaction occurs. The other important note about enzymes is that they are neither altered nor consumed in the reactions they catalyze. So they're not used up or changed. Sorry, I unplugged my computer and had to mess around with the power strip switch with my foot. An enzyme is a biological catalyst that reduces activation energy so that the reactions can occur more often, hence see previous graph. They cannot alter the free energy produced or consumed by a reaction. They can just alter the energy required to start it, if that makes any sense. So there's the energy required to start it. So my graph is all about like a downhill reaction. Hey, come back here. Stop doing things. Blah. All right, where's my pin at? There's my pin. Okay, so here's my downhill reaction. Boom. And then here's with an enzyme. Boom. Right? So you don't alter how much free energy you get out of that downhill reaction. You can just make it occur more often. And if this were an uphill reaction, boom, there's, the curve there to start it here, and you just have to continually put energy into it. And then over here, boom, you have the same energy curve of putting energy into it. It doesn't change how much energy it costs to keep the reaction going to make the total molecule. It changes how much energy it requires to start the whole thing off. Hopefully that makes sense. It also cannot convert an endergonic reaction into an exergonic reaction. It cannot make something that requires energy produce energy. and vice versa. Although I don't know why you'd ever want to convert something exotonic. Yeah. Losing track. Keep on task. So an important thing about enzymes is that they are specific to their reactions. So here's a little enzyme here, and there we go. And then let's have another enzyme here. There we go. And maybe one more enzyme over here. And you can see by the way I'm drawing this that they have different molecules to which they can bind. So maybe here's a molecule, and here's a molecule, right? Nope, doesn't fit. Nope, doesn't fit. But over here, oh, we can bind to that. This enzyme is specific to this molecule. And here, we can bind here. We're specific. See where I'm going with this. Enzymes are specific to the reactions they catalyze. They are specific to the reactants and the reaction that they catalyze. So, only specific reactants can bind to a specific enzyme. This means that for every single different chemical reaction involved in cellular respiration, there are different enzymes. So one reaction, one enzyme, because that enzyme is specific to that reaction. So when we talk about enzymes and the reactants to which they're binding, we're going to be using the term substrate. So if it's an enzyme that breaks glucose down into C3H3O3 pyruvate, then glucose is the substrate that binds to the enzyme. Glucose is the reactant, so glucose is the substrate. The substrate is the reactant. This is some enzyme terminology fun. Active site of the enzyme is where the substrate binds and where the chemical reaction occurs. So I had my little enzyme here with my active site. And the active site's essentially its shape or its conformation, three-dimensional structure, right? That determines the specificity. So, this arrow was bad there. Whatever. This is the active site. And then this over here is the reactant that binds to the active site. And this is the substrate. I'm not going to spell out substrate entirely. So, the substrate binds at the active site, and then the chemical reaction is catalyzed. So, here we go. It binds, and then the chemical reaction is catalyzed, and we break it down. Uh-oh, where'd my thing go? Aha. Boom! We break it down, and we get our 2C3H3O3. This was glucose. Ha-ha. So, substrate binds at the active site. The chemical reaction is catalyzed, and we get the products. The products do not necessarily have a high affinity. Some enzymes are bidirectional, which means the products can bind at the active site as well. But for the moment, we're just going to say, bloop, our products are released. And then our active site is now available once more. So we release our products. And there we go. And now our active site is available and a new substrate can bind, and we can run through the whole thing again because our enzyme is not altered or used up. It runs through and then it's ready again. Some enzymes can go both directions. So I have an example of that later. I can't remember it off the top of my head, Dan. But anyway, it's about CO2 management. And there's an enzyme that can catalyze CO2 being stored as, I think, carbonate or carbonic acid or something. And then that can be broken back down into CO2. So, and it's the same enzyme that does both. It can catalyze in both directions. So some enzymes can catalyze in both directions. Some enzymes do one-way reactions. So generally, your highly exergonic reactions are always one way. So if it produces a lot of free energy, if it produces a lot of energy in general, it's usually a one-way reaction. Okay, hey, there it is, carbonic anhydrase. These are both ways enzyme. So when tissue CO2 gets too high, carbonic anhydrase catalyzes the reaction of carbon dioxide and water to produce carbonate, H2CO3. But when lung carbon dioxide gets too low, so H2CO3 is inert and transported in the blood, and if lung carbon dioxide gets too low, then the same carbonic anhydrase converts H2CO3 back into CO2 and water. So we don't have a bunch of gaseous CO2 carbonating our blood, which, I mean, could be kind of cool because if you had like an enemy, you could like grab him and shake him up, and then his head would pop off, and there's carbonated blood everywhere. Oh man, when it's like 3 a.m., I get weird. I swear I'm not a serial killer. I promise. What's fascinating about this is one of the hypotheses about the reason it cranks CO2 into your lungs isn't just so that you can exhale the CO2 and get it out, but one of the hypotheses is that your brain determines breathing rates based on not oxygen levels in the blood, but CO2 levels in the lungs. That when CO2 levels build up to a certain point, it stimulates you to inhale and exhale. So that if CO2 levels drop too low in the lungs, you won't inhale. So that's a fascinating hypothesis about that. They're still collecting evidence, but there is some scientific evidence for that. Not enough that it's like a well-known theory or anything, but it's still a cool concept. So carbonic anhydrase putting CO2 into your lungs may in fact be helping you breathe. All right. So the basic cycle of the enzyme that we've drawn up already is we have a substrate, and it binds to the enzyme whenever you have an enzyme with an available active site. Whoop, boop, 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 boop. I'm getting good at that. And here, I tried really hard on that one. Okay, so here's my substrate. And here's my active site. My substrate binds to the active site, forming what's called the enzyme substrate complex. So the enzyme substrate complex is what occurs when the substrate binds to the active site. Then the reaction is catalyzed, and we break it down. Where am I? Broken down. And we convert the substrate into the products at the active site, and then the products are released. Release, release. And once the products are released, what do you know? We have an available active site again. And the next substrate can roll in and bind, and we run through the whole process again. So this is just the enzyme cycle, the catalysis cycle. Substrate binds to an active site, forming the enzyme substrate complex. The reaction is catalyzed, causing the products to form. The products are released, and then we have an active site available to which a substrate binds, forming the enzyme substrate complex. I'm not going to keep this going, but you get the idea. An enzyme not being destroyed, not being altered, not being consumed means that as long as it's present and as long as the substrate is present, we can keep the reaction going all night. So the cycle, the reaction rate, how often the reaction occurs, is based on how quickly this cycle occurs. So that's not too terrible. Most enzymes can catalyze 1,000 or more molecules per second. Pretty powerful proteins right there, right? And here is the enzyme sucrase. You'll never guess what sucrase binds to. Here's sucrase. Oh, what's this? Sucrose? Imagine that. The ASE sucrase, identifying it as an enzyme, binds to the sucrose, the OSE identifying this as a carbohydrate. So, sucrase binds to sucrose. Sucrose binds at the active site. Catalysis, so this is now the enzyme substrate complex. Catalysis occurs. In this case, we're breaking a bond, and that's hydrolysis. Add some water, break the bond, and we release the two components of sucrose, glucose and fructose. And now our active site is available. There's the cycle, the reaction cycle, and this can occur 1,000 or more times per second. So previously, this has been called the lock and key, basically where the enzyme has an active site to which only the substrate can bind, and then the reaction catalyzes there. But there's an important note I want to make about that, because a key going into a lock is like a nice, really stable structure, right? If you put a key into a lock, you actually still have to put in a fair amount of energy to turn the key to get the unlocking reaction to occur. This metaphor is breaking down. At any rate, an enzyme will actually alter the physical position of the molecule, right? So if it's trying to break down my sucrose, here's my sucrose normally, and to break it down, I have to put a bunch of energy into, breaking the cap off, sure. So instead, when it binds, the enzyme essentially distorts the molecule, right? So it alters the bond angles, puts it under pressure, kind of like taking two sides of a stick and bending them towards each other to put that big bend in it. So now that it's already unstable, it takes a lot less energy to get the reaction to occur. And the same for synthesis. It will, instead of having to take the energy to, move the molecule over, get it into position, get it on there, and put the energy into building the bond, right? The enzyme will already have that mostly in position. It'll put the stress on the molecules close to each other, so it takes very little energy. So it's not just a molecule sitting nestled in its happy little active site. The active site is actually stressing the molecules, either pushing them closer together for biosynthesis, so that the parts that are going to form the bond are very close to each other, or putting it under stress so that the bond is already stressed, so it takes less energy to break. So an enzyme alters the physical locations of the molecules and stresses bonds. So anyway, important to note, really, because the old lock and key is still a useful metaphor, but it's not accurate. Gotta be accurate, people. Okay, so as I said, the reaction rate, how often the reaction occurs, is based entirely on how quickly we cycle through binding to the active site, forming the enzyme substrate complex, catalyzing it, forming the products, releasing the products, binding to the active site, right? So the more enzymes you have, so say you've got like 50 enzymes in a solution, sure, why not? And you throw in 10 sucrose. Well, 10 sucrose get catalyzed pretty quickly. You throw in 30 sucrose, catalyzed very quickly. 40 sucrose, catalyzed, same amount of time. 50 sucrose, same amount of time. There's 50 enzymes available, 50 sucrose, catalyzed. So essentially, when it's molecules per second, right, and you have your set number of enzymes, increasing the amount of substrate essentially increases the reaction rate, or increases how often it occurs. It's getting away from me. What I'm trying to say is, with your solution of 50 enzymes, eventually you're going to put in maybe 60 substrate molecules. And then it'll have to run through all 50 before it can do the other 10. So at the point where the substrate concentration equals the enzyme concentration, we have a situation where in a given instant, every active site is bound up. So adding additional substrate will not make anything occur more often. You have to wait for the full cycle to complete, and then you have to go through another cycle. So instead of going from 10 molecules per cycle, 20 molecules per cycle, 30 molecules per cycle, 40 molecules per cycle, 50 molecules per cycle. Now, when you add 60 molecules of substrate in, it's going to be 50 molecules per cycle, then 10 molecules per cycle. Suddenly you take 2 cycles. Takes longer. So, saturation is where your substrate concentration equals your enzyme concentration, and all the active sites are bound. So increasing substrate won't get you more molecules of product any faster. Reaction rate is maxed. Enzymes are highly dependent on their local conditions. Oh, man. You're okay. Enzymes are highly dependent on their local conditions. What are local conditions? Those are things like pH, things like temperature, any like percent oxygen, amount of water, all those things constitute the local conditions. Anything that you can think of that's local to you is also a thing that could be local to a cell. And so enzymes are highly dependent on their local conditions. What that means is that enzymes function within their local conditions really well. And when you start to move in either direction away from their ideal local condition, enzyme functioning starts to decrease. So, enzymes are produced to function in a specific location with specific conditions. pH, temperature, what have you. If you get too far, enzyme, so remember we talked about conformation, right? C-O-N-F-O-R-M-A-T-I-O-N. Formation, conformation. That's three-dimensional structure. Whatever, here's a weird three-dimensional thing. Right? And let's make this an active site. Sure. There we go. Three-dimensional structure. When you get out of the local conditions, your enzyme, being a protein and having a specific conformation, remember we have primary structure, which is the chain, secondary structure, which is like the helices and the pleated sheets, tertiary structure, where you get them, ribboned up into the nice big voodoo molecule that gets its right shape, right? This is where you get something active, like an enzyme. When you get too far, we start to lose conformation. So our shape starts to get screwed up, right? We don't have that nice, super compact ribbon anymore. And when that happens, we might see our active site start to deform. Come here. And as our active site starts to deform, right, it loses affinity, right? So here's my substrate, that sphere that would have gone into the previous one well, right? It doesn't bind as well here. Maybe it'll pop out a little bit. It takes more time to get a nice strong bind to catalyze the reaction. And eventually, if you go too far, your molecule just, it permanently loses conformation. It is now completely non-functional. And the term for that is that it has become denatured. So, conformation, three-dimensional shape, denatured, loss of conformation, and thus permanent loss of function. You can't come back from denatured. You can come back from, if you're supposed to be in pH 7 and it dips to like 4 and it's not denatured, getting back to 7 will usually result in reforming the correct conformation. You'll regain functionality. But if it gets too far to say 2, it denatures. And your protein is your molecule that has a specific shape, has permanently lost its specific shape. It is now denatured and all fucked up and doesn't work. So while we're talking about active sites, so here's my enzyme. And here's my substrate. Uh-oh, what's this? My active site doesn't work with this substrate. I need something to help. Well, check it out, friend. Right here is a molecule. There. That binds to the enzyme and enhances the active site so that it performs. Now, this is a cofactor. So, this is a non-protein molecule. It binds to the active site and aids the substrate binding to the enzyme. So now, my substrate can bind and be happy because my cofactor is present. In biology, a cofactor is called a coenzyme. You can also have cofactors that are as simple as various metal ions. That can be all you need to get the correct shape, to get the correct active site. So, cofactors come in a variety of flavors. Coenzymes are biological macromolecules. You can have cofactors that are ions. So, however you want to call it. We're actually going to see coenzyme in cellular respiration, because one of the products of cellular respiration is acetyl. Oh yeah, this is turning out really good. Acetyl CoA. Oh, that's a good A. Acetyl CoA is a term for an acetyl group bound to coenzyme A. So, anyway, coenzymes help allow the molecule, the enzyme, to bind well. So, the enzyme without its coenzyme, without its cofactor, is called an apoenzyme. So when you have your poor, lonely enzyme, it's the apoenzyme, and it's non-functional. And only once you get your cofactor in there to get the nice specific active site do you get the holoenzyme that's functional. So an apoenzyme, right, is our enzyme right here, non-functional. Active site isn't specific. It won't bind, won't catalyze. And then when you have the complex of the enzyme and the cofactor, then you have the holoenzyme. There we go. So, apoenzyme, enzyme alone, no cofactor, holoenzyme, happy enzyme that's functional because it has its cofactor. And there's a much shittier drawing. All right, on to some cool stuff like enzyme inhibition. This is all about shutting down enzymes. So an enzyme that gets shut down is inhibited. So when you are inhibiting enzymes, you are slowing down how often reactions occur and even stopping the reactions from occurring. There are two flavors of enzyme inhibition we'll talk about: competitive and non-competitive. In competitive inhibition, you have your enzyme, right? And you have your substrate, right? Your substrate binds, the chemical reaction occurs, you release your products, everything's happy. But you might also have a molecule that can bind to the active site, right? So this can bind and be happy, but this can also bind, right? And then you get this structure here, right? It's still bound in. It can fit the active site, but this has no reaction. And so as long as it's stuck in the enzyme is no longer catalyzing a reaction, right? When our molecule is present, bump, the active site is blocked. No, we can't catalyze, no. So a competitive inhibitor fits and binds to the active site. However, when it binds, no reaction occurs. In effect, a competitive inhibitor competes with the substrate for the active site. And how much inhibition occurs is going to depend entirely on the concentration of substrate versus the concentration of inhibitor. So, if you have more substrate than inhibitor, you're probably going to still see the reaction occurring, but it'll be slower. If it's 50-50, you'll expect to see about half-speed reactions. And then as you get more inhibitor present, you get slower and slower reactions. And then if you get to a point where all enzymes are bound up to an inhibitor, you have completely inhibited the enzyme and the reactions stop. Penicillin. Oh man, that is an awful P. Penicillin is a class of antibiotic we discovered in a fungus. And it is a competitive inhibitor. What it does is it competes for the active site of an enzyme that builds the cell walls of a bacteria. So your bacterium has its little membrane, and then it has a cell wall surrounding it. A certain class of bacteria has a really thick cell wall, and it needs that cell wall to survive. And so penicillin competes to inhibit the enzyme that produces the cell wall or catalyzes the production of cell wall. So if you get enough a concentration of penicillin, you'll block up the enzymes on the bacteria. They can no longer make a cell wall, and they can't replicate, and they all die because they're sad. So competitive inhibition, pretty cool stuff. Now let's talk about non-competitive inhibitors. So again, well, no, I want to be over here. So here's our enzyme. Oh, this is a weird looking one. And here's our active site. Active site. Here's our substrate. And now we're going to talk about something new. This is called the allosteric site. Oh my God, this is going to take T-E-R-I-C. Oh gosh. The allosteric site. Oh, looks so good. Allosteric site is a binding site that has nothing to do with the active site at the moment. So in non-competitive inhibitors, we have the presence of an allosteric site. And here's my allosteric inhibitor. Oh, that's close enough. Allosteric inhibitor. It can't bind to the active site, right? Nope. Sorry, doesn't fit the active site. Only the substrate fits into the active site. However, our allosteric inhibitor can bind at the allosteric site. And what happens when it does this is pretty cool. So here's our allosteric inhibitor. Here's our enzyme, and it has now binded at the allosteric site. All right, so here's our enzyme before, and here's our enzyme with the inhibitor. And look, our poor substrate, oh no, I can't bind anymore. So an allosteric inhibitor binds at the allosteric site and changes the conformation of the active site. And when it does that, it prevents the substrate from being able to bind. So we have an allosteric inhibitor that binds at the allosteric site. Once it binds, it alters the conformation, changing the active site so that the substrate can no longer bind at the active site and we cannot catalyze. So, boom. Allosteric inhibitor straight switches an enzyme off. So here we go. Here's our inhibitor here. Here's our substrate. And here's our happy enzyme on its own with its active site substrate binds in, we have our enzyme substrate complex. But when the inhibitor binds, we change the active site, and now, oh no, we can't bind. Oh, so sad. Let's see, cool inhibitor information. So cyanide, potassium cyanide, the stuff you drink if you're a spy that's gotten caught, is a allosteric inhibitor of an enzyme called cytochrome oxidase, I think D. It's basically involved in the very first step of cellular respiration, and it irreversibly shuts off the enzymes involved there. And so once you shut off the enzymes involved at the very start of cellular respiration, you shut off cellular respiration. So cyanide kills you by being an allosteric inhibitor of the enzyme for respiration, or for starting respiration. Anyway, cool. Allosteric sites aren't just for shutting down enzymes and killing you. In fact, allosteric sites are a natural part of many enzymes, and they're there for a reason, and that reason is allosteric regulation. So a cell does not necessarily need an enzyme to be constantly catalyzing the substrate whenever the substrate is present. Sometimes you don't want that. Maybe you're trying to build up a specific concentration of substrate. So if the substrate concentration dips too low, you don't want the enzymes to continue catalyzing that substrate. So you turn it off. So you have an allosteric switch. And since we're talking about the possibility of, turning off an enzyme if substrate concentration gets too low, there are certainly enzymes that we can switch on if substrate concentration either gets too high, or in the case of some bacteria, there's an enzyme for digesting a specific sugar, lactose, and that enzyme is normally turned off. And if lactose appears in the environment, they can turn it on. Anyway, so basically an enzyme can either default to inactive or active. Enzymes that are allosterically regulated will default to either inactive or active. So an inactive enzyme can be switched on by an allosteric activator. And an active enzyme can be switched off by a repressor, an allosteric repressor, an allosteric inhibitor. So if I'm talking about something dangerous like potassium cyanide, I'll say allosteric inhibitor. And if I'm trying to talk about regulation, I'll use the term allosteric repressor. Because cyanide is an inhibitor that irreversibly stops. An allosteric repressor can be removed by another enzyme. Anyway, cool stuff. Good Lord, I think that's the end. Yeah, there might be one more slide. Okay. Is that it? Yeah, okay. Yeah. So here's my picture of various of the types of regulation. So here we have a substrate, and it can bind and produce our products, and our enzyme is defaulted to active. So we want to turn it off. So here's our allosteric repressor, binds. We have an altered conformation, and our substrate is unhappy, can't bind anymore. Here we have an enzyme that's inactive, but a substrate arrives. Oh, we need to digest that. So here comes the allosteric activator. It binds, and now we have a conformational change, and we have an active site that will actually work. And so our allosteric activator turns the enzyme on so it can catalyze the reaction. Our allosteric repressor turns the enzyme off. So if you're default to inactive, then an allosteric activator turns you on. If you're default to active, then an allosteric repressor turns you off. So, like men with mustaches are an allosteric repressor for you. I don't know. Feedback inhibition. Okay, so feedback inhibition is another neat thing involving allosteric sites. And what's unique about feedback inhibition is that you have a non-competitive, an allosteric inhibitor that shuts down an enzyme. But that allosteric inhibitor is actually the product of the reaction the enzyme catalyzes. And this is all about homeostasis. So here we go. Here's our ideal level, right, of whatever. And so an enzyme is catalyzing. Woo! But if it just keeps catalyzing, we'll go beyond the ideal level. So at the same time as we're catalyzing and getting our products, that products starts binding to the enzymes. Down here where we have a low product concentration, we're not going to get a lot of enzymes bound up by the product. But as the concentration increases, we get more product around the enzyme. The more product we have around the enzyme, the more enzymes are likely to get shut off. And so as we increase our reaction rate decreases. And if we bind them all up, it may even stop. So feedback inhibition is where the product of the catalyzed reaction binds at the allosteric site and acts as a repressor, which is pretty cool. It's just sort of a baked-in homeostasis mechanism. And then if the concentration is too low, you have other enzymes that catalyze the removal of the product from the allosteric site, reactivating the enzymes. So feedback inhibition is pretty damn cool. It's a pretty heavy influencer of homeostasis, of a lot of chemicals.